It's time for another episode of CRISPR Corner. In this episode, we're going to talk about the different formats of CRISPR reagents available and how to choose the best one for your work. There are 4 main formats that you can use to introduce the CRISPR machinery into your experimental system: DNA, RNA, protein, and virus. With DNA, your Cas9 and guide RNA will be expressed from a plasmid that is introduced or transfected into your cells of interest. Plasmid DNA is easy to produce, manipulate, and handle in the lab. Many commercially available CRISPR plasmids are optimized for transient transfection and are diluted out of your cell population with each cell division. This can be a pro if you don't want Cas9 active in your cells after your intended edit is made, but could also be a con if your cells are rapidly dividing or the target genomic site is not readily accessible. CRISPR plasmids can be all-in-one, meaning the Cas9 and guide RNA are expressed off the same plasmid or separate. The plasmids can also contain fluorescent markers to help you visualize which cells received the CRISPR machinery. An important consideration to keep in mind when using the plasmid format is that the optimal promoter for your cell line may not be the promoter used in commercially available plasmids. It's always a good idea to scan publications that have used CRISPR in your cell line of interest to see what has worked previously. One final consideration for plasmids: there is some risk that your plasmid DNA could randomly integrate into the genome. Overall, plasmids can be a great option, especially if your research is cell culture based. Now let's talk about RNA formats. These are inherently less stable than their DNA counterparts, which makes them harder to produce and more difficult to work with in the lab. RNA is also degraded quickly in the cell, which means that if your cells have a long doubling time, RNA might not be the best format for your work. However, RNAs are also promoterless, leading to less variability in activity between different cell types, and they have virtually no risk of random integration into the genome. Overall, RNA formats can be a good choice for embryo microinjections and in specific cell types. Moving on to protein. As the name might suggest, your guide RNAs are not going to be a protein, but your Cas9 can be. In this format, your guide RNA can be either a 2-part CRISPR RNA or cRNA plus trans-activating CRISPR RNA or tracer RNA, or a synthetic single guide RNA. When you combine your guide RNA and Cas9 protein, you make a ribonucleoprotein, or RMP. RMPs are a great option for slowly dividing cells or cells that exhibit toxicity to exogenous DNA. RMPs also show rapid editing since the components do not need to be transcribed and/or translated. Overall, RMP is a great choice for many applications, including microinjection for animal models and transfection in cell culture. The last format I want to discuss is virus, and specifically I'll be discussing lentivirus. Lentivirus is ideal for screening applications as it will give you the highest editing efficiency for any sort of editing screen. Lentivirus can also be a great option for difficult to transfect cell lines and/or slowly dividing cells. Lentivirus will randomly integrate into the genome of your cell line, giving permanent, continuous expression of your CRISPR machinery, which may or may not be what you want for downstream applications. Like plasmids, lentivirus vectors can be all-in-one or have 2 vectors, one for your guide and one for Cas9. To add a level of complexity, you can also mix and match some of these formats. For instance, delivering your guide as a plasmid and your Cas9 as an RNA. All this to say, there are a lot of options when it comes to deciding how to introduce CRISPR-Cas9 into your research. In the end, your decision is going to come down to your research goals, your experimental systems, and what techniques you're most comfortable using. Each experiment is unique, and you may find yourself using different formats for different projects. If you'd like to discuss which format is the best fit for your upcoming projects, reach out to your local Millipore Sigma team. We have a team of specialists with the job of helping connect you to the CRISPR technology that will help accelerate your scientific discoveries. That's it for this episode. Next time on CRISPR Corner, I'll be discussing options for screening with CRISPR. Until then, catch up on previous episodes and reach out to your local Millipore Sigma team if you'd like to discuss how CRISPR might fit into your work.